The next presenter is Zach Maloney, who's a graduate student in the lab, who's going to share some of his background on what he's doing for our experimental work in the lab, and what we're planning to do in terms of imaging, also to augment our cognitive testing. So Zach?
Hi. So yeah, as Dr. Kudub said, I'm a graduate student in her lab, and I'm going to be focusing a lot on sort of the more direct biological ways of looking at your brain. So our big problem is we would love to be able to look directly at the brains of our volunteers, but that's difficult. The brain is behind this nasty thing called the skull. It's kind of rough to pull out neurons from your brain. If we want to image it, we'd have to put you in an MRI, which takes time and it's a big clunky machine. So what we have to do is think of ways of looking at the brain without looking directly at it. And a really good way of doing this, which people have done in the past, is by looking at your retina. So when you go into the ophthalmologist's office and they give you that eye scan, right, where they put the bright light in your eye, sometimes they do it manually. Nowadays, they do it with these machines, what are called fundus imagers. Literally what they're doing is they're taking a picture of your retina. And this is useful for them because they want to see if there's anything wrong with your retina. But it's also useful for us because it lets us look directly at the blood vessels in the retina. And these are really the only non-invasive way that we can look at your blood vessels. And they're so close to the brain that the structure of them changes. Well, what they do changes as the brain's blood flow also changes. It's basically a way of looking at the blood vessels that look like those in the brain. So why is this important for us? Well, we want to look at a couple of things, right? We want to look for leakage. We want to look for changes in blood flow. We can use this great imaging method called OCT, which allows us to actually look at the full thickness of the retina and look at those blood vessels buried underneath it. So everybody's structure and everybody's flow dynamics are going to be different. So maybe we want to pull out features like branching, leakage, blood pressure mapping. And these things have actually been used in the very recent past to correlate with diabetes, shows up when you do this. You can even correlate this with mild cognitive impairment. So if you look at the retina of somebody who has cognitive impairment, it's quantitatively different in terms of its dynamics than somebody who doesn't. So cool, people have done this kind of point in time, but what is it that we can bring to the table? Well, we have our great volunteers who let us do this multiple times, who are letting us track their behavior.
So I'm sorry, Zach has been having internet issues this morning. He just moved into a new place where they're setting up internet. So he's summarizing what Zach had been sharing. What we plan to do over time is to look at retina scans before and after interventions, and also periodically, so yearly for those volunteers who want to participate in this, in parallel with cognitive testing and the wearable device data we're already collecting. At the same time, the next slide actually goes into more details about what we're doing in the lab, using neural-based models that derive from the blood of the volunteers who we're tracking systemically through these non-invasive scans. So once Zach gets on, I'll let them explain it, but as I don't want to delay the workshop, so I'll go ahead and go to the next slide to give you a little bit of overview about what we're planning to do in the lab. So this slide shows a cool project, which is actually Zach's thesis project that we started planning this past year, and which we'll start to be able to do once we're all fully back in the wet lab. And this is the idea that we can make these models of the brain, and particularly models of the brain in a region which is regulating sleep, that are derived from the same cells that we collect from our blood samples periodically from our volunteers. So how do we do this? So from the blood, you can get what are called peripheral blood mononucleated cells. And these cells contain the same genetic backgrounds as other cells in your blood and your body. And we manipulate them or change their behaviors and their function using what are called Yamanaka factors. These are genetic factors or factors which manipulate the expression of different genes in your blood cells. And that allows us to actually make neurons. And not just neurons, neurons that are a particular subtype and allow us to form a region of your brain as a mimic in the lab. And the region of the brain that we're focused on is the suprachiasmatic nucleus area, or the SCN. And the goal of that is because the SCN is really your clock of your brain and your body. It regulates cell cycle throughout your body. It regulates, circadian rhythm throughout your body. And it's thought to be responsible for your wake-sleep cycle, and also a main area of your brain which is changed or abnormal if you have a sleep disorder. So the hypothesis there is that we could use small molecule inhibitors or stimulators to also test out theories of how your brain might change in this particular region that regulates sleep. And then also in turn, see how people's brains are really changing, how their sleep patterns are changing, and their behavior is changing, while we're parallel working with these experimental models in the lab of the region of the brain that mimics sleep. So Zach, if you're back on, it doesn't look like Zach's back on yet, but we'll give him the chance at the end when we have the Q&A to follow up on this. This is really exciting work. It's extremely outside of the box because we're really making models of living systems of volunteers in our project and specific regions of the brain that are regulating sleep and behaviors that we can track, that are ongoing behaviors we're monitoring using wearable devices.